Okay, so let's start. Thank you very much for coming to another seminar. Today we have a special seminar, as you have seen in the poster and in the announcements, because it's not a usual seminar. Because today we have three presenters that are people that they have enjoyed a JAE Intro scholarship. That is a scholarship that PhysIC gives for master students, so they can work in a research center while carrying out their master thesis. So the people that enjoy this are enjoying this scholarship during this year is going to make these presentations this Friday and the next Friday also. So today we have the first three people that are Esther, Eva, and Chiara. And so each one will make a short presentation of around 10 minutes, 10, 12 minutes about their research here at IMEDEA. And what we will do is after the presentation, there will be a short period of questions for the person until reaching the 15 minutes for each of them. And if anyone that they don't have time at the end, we also will leave final question time after the last presentation. So this way, each one will have their questions. So they are master students, so also enjoy the talks and then try to make nice and constructive comments because they have done a big effort to present today. So the first one is Esther Ivars. So yeah, the floor is yours.
Thank you. Okay. Okay. So good morning, I'm Esther, and I'm going to present the research that I have been done here in the IMEDEA, thanks to the JAE Intro grant from PhysIC. And I title my presentation, my research, Biophysical Aspects Affecting Substrate Detachment of Benthic dinoflagellates. First, I want to do a brief introduction. Dinoflagellates are a diverse group of unicellular protists that in the recent decades has increased the interest of the scientific community because of the recurrent episodes of harmful algal blooms that obviously are has negative impacts in the environment, but also can be toxic for humans. Specifically, the benthic dinoflagellates live in close relationship with the substrate, so there has to be specific adaptations to live in these environments. Some authors talk about mucilage production that as a mechanism of adhesion to the substrate, but also to act as a physical barrier to protect the cells, and can contain chemicals that can be their own toxins or maybe to trap nutrients to facilitating the absorption. I want to introduce two aspects that is that my research is based, that are microbiology, that is a study of the behavior of fluids at microscopic levels, and in short, the production of this mucilage increases the viscosity of the surrounding environment, so this affects the ability of the dinoflagellates to interact with this environment. The microbiology based on the laminar flow because of these microscopic dimensions, and in this specifically in these microscopic dimensions, the number of Reynolds is too low, lower than 1. So the flows that predominate in these environments is the laminar flow. In the other hand, microfluidics is the science that studies the manipulation of fluids in a microscopic scale, and is also based on the principle of the laminar flow because of the microscale dimensions of the channels in the chips that we use for these techniques. So any change that we do in the flows we applied can be monitored and controlled with precision. The cultures that I use are from Colwellia, that is a benthic dinoflagellate. And the objectives that I proposed for this research was to optimize the nutritional conditions for mucilage production in this species, also to analyze the rheological properties of this mucilage, and finally to evaluate the response of a biofilm of Colwellia to different flow rates that I applied using microfluidic techniques. For the experiments on the different natural limitation, I use a 96 well plate to obtain 32 replicates for each treatment. The treatments were control and nitrogen and phosphorus limitation. I recorded videos and also photos to analyze them in the software Fiji to obtain parameters as the number of cells, the ferret diameter, the circularity, and also the biovolume. For the microbiology experiments, I use a solution of fluorescent microspheres that I inoculate in each well that I want to sample. And again, I recorded some videos that look like this one. And the script that I use for recording these videos calculates the diffusivity of the microspheres in the environment. And this allows us to obtain the parameter of the viscosity of the environment where the microspheres were. And we can calculate the relative viscosity, taking into account the theoretical viscosity of the seawater. Finally, for the microfluidics experiments, I do first of all a control with polyethylene microspheres of 20 micrometers of diameter that is more similar to the diameter of my cells. I introduce the microspheres into the channels of the chip, and I apply a range of flow rates. I also recorded videos to analyze after that the parameters that I have interest, and specifically I want to know the percentage of spheres that remains attached to the bottom of the chip. So I use a script to remove any inconsistencies between the frames of the video. So only it remains the spheres that remains attached in each frame of the video. So after that, I analyze the images with Fiji and obtain the number of the microspheres in each photo for each flow applied. Finally, with Colwellia cells, I do the same. I introduce the cells inside the channels of the chip. I wait for 3 days because of to improve the creation of a biofilm of these cells. And I also analyze by one hand the number of the cells that remains attached in the bottom at the same way as the microspheres. And in the other hand, I would like to analyze the trajectories of the cells, because when I was recording the videos, I saw an interesting behavior of the cells that they actively swim when the flow rates are moderate. So I want to try to maybe to explain this movement, and I do the track of the cells, and after that I calculated the Riemann square displacement, that is a measure to quantify the displacement of the cells in time. Regarding the results that I obtained, I do not see any differences, significant differences between the growth rates, but regarding the cell size, I observed differences between the exponential phase and the other phases of the cell growth in the ferret diameter and also the biovolume. And in circularity, I identified gradual decrease of this parameter, and also the differences observed were significant for each phases. Due to the cells densities that I obtained during my experiments, because I have a little bit of difficulties in the lab to reach more abundance of the cells, I think that the nutritional conditions that I used were not sufficiently restrictive to my cells. In addition, some studies have identified the ability of some dinoflagellated species to maintain their cellular structure and this maintain the functionality of the cell. Okay, for the results in microbiology, the map that we obtained after the script allows us to identify where the viscosity is higher in the area that we take photos, and we identify the more viscosity areas near the cell walls, and also in the areas that there is a mucus loose in the medium. Regarding to the relative viscosity, the highest values of relative viscosity were obtained in the phosphorus limitation treatment, and also these differences were significant. I suggest to explain these highest values of relative viscosity in the treatment limited with phosphorus because of the overflow mechanism that has been explained by a lot of authors, and they explain that under phosphorus limitation, the cells blocked their cell cycle, and devote their efforts to accumulate and create and accumulate the polysaccharides that maybe can be I don't know how to excrete to the media, so this maybe can increase the viscosity of the surrounding environment. In contrast, under nitrogen condition limitation, the cells prefer to reduce the protein synthesis and to adjust their metabolism and not to accumulate the polysaccharides. For the results in microfluidics, the control shows that the microspheres of 20 micrometers of diameter were disadvantage of the bottom at more or less 250 micrometers per second, aligning with the physical forces that are affecting these microspheres. So this allows us to establish a reliable framework to the next experiments. For the Colwellia cells, we saw at this graph that in addition to withstanding much higher flow velocities than microspheres, the detachment rates were lower also. This indicated to us that in addition to the physical forces acting on the cells, there must be also some biological mechanisms that is favoring the adhesion of the cells and flow resistance. Finally, the results for the rhythmics for displacement. It appears that during the moderate flows, the rhythmics for displacement values are higher, maybe, and this is more clearly to see. And above 4,100 micrometers per second of flow velocity, the cells decrease their rhythmics for displacement values. So it is like cells starts to swimming a lot until 400 micrometers per second, and after that they get stuck in the bottom of the chip. Okay, so as I mentioned, the cells were resisting the applied fluxes to a greater extent, suggesting that they must be applying some biological mechanisms add to the physical forces that are affecting the cells. Some authors, especially in bacteria, have described mechanisms of mobility and secretion of exopolysaccharides as a mechanism to resist drug forces. As Colwellia does not have these structures like these, I can explain it so clearly, but the swimming behavior observed in experiments could suggest an analogous mechanism. And after that, in relation to the root meniscus displacement, I only found a study in dinoflagellates that it talks about Caenia brevis. And it has shown that this dinoflagellate adjusts its flagellar motility to remain resuspended under different currents in the natural environment. So maybe it could open a new research line to explore this mechanism. So a summary of the conclusions that I obtained is I want to highlight the relationship between the mucus production and the limitation of phosphorus. I propose to repeat experiments with higher density of the cells, also to improve the better growth of the cells, but also can benefit the microbiological analysis. And I propose to do some genomic and proteomic analysis to analyze the metabolic pathways that are being activated under these conditions. And in relation to the dynamic behavior of Colwellia under these moderate flows, I propose to do more experiments to can be able to suggest a pattern of the movement. And a future direction that I propose is to compare the laboratory results with actual flows near the shore boundary layers, because of here affects also the turbulence and the currents and variations in the velocity of the flows. So maybe can affect in a different mode in the laboratory. So thank you so much.
Thank you very much, Esther. Very super nice presentation. So we have a couple of minutes for questions for Esther. So, anyone?
Okay.
No, Marco, please. No, so, well, super nice presentation, thank you. I was wondering about the part of the experiment in which you see how the motility changes with the increasing flow rate.
Yeah.
And I was wondering if in the part of the life cycle of the organism, is there anywhere a part in which it actually swims? Because as I understand, it's usually like just stay attached to the benthos, and then from there does all its life, it can remain there.
Yeah.
So it's a cell that has the possibility of swimming, but it usually doesn't swim in its normal.
Yes, I think it will be a relation with the cell cycle, the life cycle. I think it depends, because in my cultures, when cuando agitaba los cultivos, sí que es verdad que las células nadaban bastante más que cuando dejaban los cultivos ahí tranquilitos. Sí que tendían a generar este biofilm pegaditas al fondo. Sí.
And I was wondering, so, I mean, the interpretation of the result at a certain point, it just goes sediment, is that because the flow is so strong that it overcomes like their possibility of swimming and they give up. Like you put a flow on them and then they detach because, well, at a certain point the flow is too strong and they detach. So they start swimming, and I'm wondering if they try to go back. I mean, why do they swim? Like, do they swim trying to go back to the benthos?
Sí, no he podido saber muy bien por qué lo hacen, pero creo que sería súper interesante seguir con estos experimentos para poder concretarlo un poco, pero sí, creo que pueden estar, aparte de nadando un poco a contracorriente para mantenerse en el área, buscando una zona donde poder
Para tener
anclarse.
turbulencia.
Sí.
Me pregunto que si llega un punto en el que simplemente el flujo es tan alto que se desflagelan, porque igual pierden la colita y
No,
ya...
la verdad es que estaban aguantando, bueno, creo que está aquí, estaban aguantando flujos hasta 32.000 micrómetros por segundo, que es lo máximo que daba ya la bomba de sí, y ahí estaban las tías bien agarradas. Sí que es verdad que muchas se terminaban soltando y quedaban arrastradas por el
Claro,
flujo,
sí.
pero muchas, una gran parte se quedaban ancladas al fondo. Como que al principio sí mostraban este movimiento más aleatorio, y conforme seguíamos subiendo los flujos, se quedaban pegaditas y ahí estaban. Nadie resultado.
Vale.
Creo que esto, o sea, puede ser muy interesante seguir viendo porque realmente no he podido encontrar una explicación muy concreta sobre este mecanismo, así que creo que puede estar guay mirar a ver qué podemos sacar de ahí.
Marco, you want?
No, I'll ask you later.
Okay.
Okay,
Okay, so we can leave it here. So thank you very much.
thank you.
And the next speaker is Eva.
Welcome everyone. My name is Chiara and I'm a master student at the University of Padova in Italy, and my supervisor is Joan Pons. And I will talk about phylogenetics. So Tick-tock, tune the clock, ongoing search into Peracarida's evolutionary timeline.
Here, once.
Okay. Briefly, an overview of what I'm talking about. I will start talking with an introduction about phylogenetics. What is it, for make a context, if you don't know. So we can start talking about tuning the clock, what does it mean, and implication. Finally, I can finally start with the global phylogeny and molecular clock, show the focus of my work here, and I can show you my trees, my only result, so be careful. It's really important. And finally, a summary of everything, so you can keep it in mind like a take-home message. Okay, briefly, an introduction. Phylogenetics means the study of evolutionary history through genetics. And here you can see the classification of taxonomy. So we have species level, which is the more specific one, and the life. And this is a representation of phylogeny, which is a tree. The recent common ancestor is here, it's the more brittle one, and then branches and nodes with the edge, which are the tips, with the recent species, so the nowadays species. And this is the subject of my work. I'm studying Peracarida, which is a super order, and there are some pictures of in specifically, my study is on about the order Amphipoda, and I will concentrate on 3 families, Iaiellidae, Metagargoniti, and Pseudonipalgidae. This is my tree, as you can see, and here you have the cluster, but I will explain it later. Now I want to show you a really cool thing, which is the One Zoom platform. And if I can do it. Yes. Okay. It's a really cool visualization. And this is the tree. I don't know how to use it. Wait. Oh, see. But for example, if you wrote, for example, Homo here. You can go back all the time and start again. So this is a school course software. I recommend it to use it. And yes, this is us. And for coming back. What should we do?
You want to go to the presentation?
See,
Yeah.
this one. Okay, here we are again. Well. But this is a tree. How can we build a tree? A tree, it's based on the evolutionary distances. They are the changes that you find the mutation inside two lineages. So, for example, we have a tree based on genetics, and here it's longer than this one, because here we have more mutation than in this small branch. And this set the relative time of the tree. What does it mean with relative? Because it's just saying the events, some events happen first than others, but they are not set to our time. It's just the time of the tree. But we want to calibrate it, and we really want to say, okay, this tree, how can we locate it in the story of the heart, for example? So there are two main questions, and this is really the focus of my project. How can we calibrate a phylogeny, and how can we tune the clock? How can we synchronize the relative time? Well, this leads us to the global phylogeny, which is an intricate context of parameters. And I show you different years because are some features that are really well interconnected between each other, and some of them can really move the clock behind. So we have the data here, the clock model, age constraint, and branching method. The first one is the data. They are all the information that are connected to our data. So the sequences, like this one, they are string of information with different nucleotides, they are 4, and also the mutation and the substitution model, because we know that mutation between the 4 nucleotides, they are not all the same. They are different in weights and frequencies. So we have different models to explain this. Another really important features or gears, as I call it, it's age constraints. So this means we want to calibrate our tree, but we need fixed point in the story of the heart. And we can use fossil age or biogeographical events. I want that you notice this gear here, the clock model, which is a connection between relative time that we have from the data, the relative time from our data set, and the real time, the absolute age. They are connected by the clock model. So it's the method we use to set and to tune the clocks. Lastly, there is the branching method, which is a bit marginal in my study, and just explain how diversification and speciation are happen inside the tree. They are two models, and these two models, one, the Yule one, it's the more simple one, because you have species and they just diversify and split, but never extinct, which is inside the other one. Okay, let's focus on the clock model because it's the one I'm really trying to adjust. Okay, the clock model try to explain the mutation rate. So the connection between the relative time, the absolute time. So the rate at which mutation occur, our lineage should match the time of our, normal time. So we have different models to explain this, and we can categorize them in two important boxes. One is the strict clock, the other one is the relaxed clock. The strict clock assumes that in every branch you have the same evolutionary rate. So there are no changes inside the branches. But of course, this is very controversial. Reality, it's not strict, unfortunately. Or maybe not, because biology is very beautiful for this way. So the best one is the relaxed one, but there are different kind of relaxation. In our previous studies, we use the uncorrelated and log normal clock, which means, okay, let's start with the correlated. Correlated means that you have different branches and different evolutionary rate, but in some ways in the same clade, so in the same branching from the start one, they are correlated, so more or less the same one. Uncorrelated, it's the opposite, they are not correlated. And you can assume they have two different types of distribution, log normal or exponential. Another really interesting one we are trying to focus is now, it's the mixed correlated and uncorrelated clock. We try to, in some way, balancing these two clocks together. Yes, it seems that in Peracarida, we are trying to use the uncorrelated log normal and doesn't match the absolute age. What we do, what can we do? So we are trying to use the mixed correlated clock. What I'm talking about? I'm saying that if you have a tree, we are hypothesizing that you have different clock in each branch. So how can we resolve this? It's really complicated, really intricate, also because we don't know exactly the evolution rate, we don't know the timing. How can we move inside this rooted and intricate branching? This is the major question, and I think it's really astonishing if you think about it, because it's very difficult for me also to find this image and to show you. So, yes, this is Joanne. I think he has a bit less hair right now, but because he discovered before. Well, so what can we do? How can we manage this problem? Please try to implement these two new clock, mixed effect and shrinkage effect clock. Right now, I don't have results to show you. I don't know if it works or not. I really hope to. I hope to have results. But basically, the mixed effect clock try to balance a bit the correlated and uncorrelated clocks. Meanwhile, the shrinkage, it's more or less a slow modification of the clock. So it starts maybe in a point and assume that slowly there is a shift of the evolutionary rate. Well, my result. It's a preliminary result because it's not set the time, it's not calibrated. It just show the relationship between species, and you can, but it's really interesting for me. And I want to show you a few things about trees in general. First things to see, our group. You know species that are at the real base of the tree. So they are used and they are really important to set the root, the starting point of the tree. They should not be forgotten in a tree. Otherwise, you will not be able to, correctly locate the other species that maybe are more interesting for you. So you use them and you know they are there, they are the edge. It's like a framework. Secondly, Isopoda order. This is the antipode order. Isopoda, it's really important in my case, and it's here, because we have fossil record. So we have dates. We can calibrate it, actually. I didn't have done it yet, but it's an ongoing process. Lastly, I want to show you the three families that are well clustered. So they are not mixed, they are perfect there. I don't know exactly the age of them, but they are there and you can see them. Last thing that I really want to show you, this is another formation of my tree. These are the booster values. They are important in phylogeny because they are the confidence of the node. As bigger it's the yellow circle, as less it's the confidence. So actually, we have no idea what's happening here. We know that there are a cluster here. They are sister group. This is okay. And here, we don't know. And it happens sometimes, but it's okay because we don't have the power to know everything about phylogeny. At least we know that they are clustered really well. And here, we have to study more. We have more samples. We need more samples. We need to go into the details and to discover. But this is the power of the phylogeny. It's not completely known. Well, as short summary, so if I lost you, we can find us out here. Molecular clocks are essential tool for estimating the timing of evolutionary events. So we can see in the past, but they rely on precise calibration point to yield divergence estimate. However, sometimes branches can have different mutation rate. This is very important. Take home message, highlight. Branches can have different mutation rate. So ad hoc tuning of a molecular code is required, but it's really difficult to do it and difficult to implement it. There are many parameters. I didn't show you the background. It's a mess, but really it's important to do it. And lastly, Peracarida's timeline remains a mystery. So we don't know. And stay tuned for the following. Joan will do a verse. Okay, thank you very much for listening.
Thank you very much. Yeah, super nice presentation. Any questions?
Data.
Have a very, you know, curious.
Sì.
I'm wondering when you consider the different rates of evolution and branches, do you also take into account the ecology of the ecology?
See, actually, it's really important. Thank you for the question. I didn't have a time for it to say, so I'm really happy to let you say. This is the starting point of my investigation, because, for example, we have 3 different families, and they are really, I mean, there are some species inside the Mediterranean Sea, and another one in the South America. I don't remember the exact definition of this different pattern of distribution of the species. But also, some of them are living in caves. So in an environment in which you have a different evolutionary forces inside it. So of course, this influence the tree. It's really important to take it into account. And I didn't have a space for put it in with my ideas. But I think here should be put also the context, the environment concept of a species for build a perfect global phylogeny. I didn't for now, because with just the DNA, you don't take into account at the beginning the environment content. But I think in the end, you should take it into account to have a good explanation of the framework. I hope to answer the question.
I can make I'm wondering if things like the lifespan or the growth rate or some allometric relationship with the size and the metabolism.
See. I think so, but you have to ask yourself if at which level you are watching the data that you have, because I'm watching really long in time. And for example, maybe this is interesting if you watch a species in a shorter frame of time, and this makes a difference. But if you watch, for example, Peracarida in a wall, they are more or less homogeneous under this point of view. And I hope Joan can confirm what I'm saying.
Yeah, this question is more important to answer afterwards, when you have the phylogeny. And when you have the phylogeny, you can ask this question and you can reconstruct if the ecology can explain the different rates you find. For instance, you find that all the species that live in cave, they have very higher rates than the one living outside. You could explain why the rate would be related to that. But at the beginning, you cannot use any of this information to build the phylogeny.
Also because you will be
You can answer
biased.
many questions when you have the phylogeny. The phylogeny is a framework to analyze any character. You can analyze if the swimming have been appeared 3 times in the family, or appears only once in the life, or you can reconstruct any character on the phylogeny. But all these characters is very difficult to use it to build the phylogeny. You can answer many questions, of course, but to build it, you can have it in account to answer a question afterwards, not before.
But I'm wondering if you'd be very interested, you know.
No, it is very interesting. I mean, in a sense, the phylogenies are given for two reasons.
No, but
To build the systematics and to know how evolves characters through time.
what I'm wondering is if you know a priori, for example, about how many generations per on time you can have.
Yeah,
That
true.
that should
That's true.
that
That's
should
true.
affect the rate of evolution.
Yes, that's true.
Maybe some of this could go.
For population genetics, yes, it does. That's very important for population genetics. But in this level of phylogenies, it's so changed so much that it would be impossible to look because for most of the species, we just know that they exist. Lucky us that we know that exist. We don't know about anything about ecology. Because the one that live in caves, they live in caves, and you have to find them. Sometimes you have to go with the fellow driver. That she will lay in the cave and we don't know about anything about ecology. And in bacteria, most of the time they don't know that they are there and they don't know much information. I mean, we know more. These things are built more to know the diversity that to because we don't have any information about the other things. Would be interesting, but we just know that they exist. It's very important the population size and the time of the time that you have generations. That's very important for population genetics. That is very important. For the phylogeny, I think it's not because they change so much from one to the other that it's difficult to compare.
I have a question, more of a curiosity. For to build your phylogeny, did you stick to one methodology like parsimony versus likelihood?
Wow.
Or you are of like a one credo, or you explore it very
Thank you again for the question. Actually, I prepare more slides. I can start again. Well, I don't know if I can present it from here. But can you see? Yes. Okay. Quickly, really quickly. This is the upstream, what I have done. So I have a sequence, the data, I clean them and I prepare them, and I prepare the dataset for the phylogenetic analysis. And then you have two different frameworks for doing this analysis. All the gears that I explain you can do it in two different types of algorithms. The maximum likelihood and the Bayesian inference. They are different. We are using, at the beginning, the tree that I show you was from the maximum likelihood context, but now we are working on the Bayesian inference context. They are different, and this is more precise in a broad sense, because maybe from the maximum likelihood, you have one single result, but maybe it's wrong. It's just, maybe you are in the local optimum of the context of the tree. You don't know. In here you have maybe more parameters, and the curve of the algorithm is a bit larger, but it's more precise. So yes, the first tree was the maximum likelihood one.
So you got the different results of the
I don't have results too.
No, the result more or less the same, topology and.
Should be the same.
In Bayesian, the good thing you have the uncertainty. I mean, in the sense, the maximum altitude is giving one answer. And to find the uncertainty of that answer, you have it in Bayesian, you have the uncertainty in the topology, but you have also the uncertainty of all the parameters. For all the parameters, you have a distribution, it's not a single answer. And it's more, no more precise, but it's giving you more information about how is the variance in each one of the parameters.
It's more reliable.
And also Bayesian is much lower, and like maximum altitude is very fast. I mean, Bayesian is no way to build a phylogeny for more than 100 or 200 taxa.
Yes.
And maximum altitude you can build with thousands of nits. It's different approach, but that's.
There was one last question.
Right, so when you're building a phylogenetic tree, there's the classic way, which is based on similarity of structures, right? And then there's the new way based on DNA. And basically what you're doing is trying to find out the order of evolution there. So here you centered on the DNA way and sort of ignored the other one. Can you think of any case in which the classic way is more correct than the DNA?
With classic way, you mean morphological traits?
So structures, say,
Structure.
so you see a bone appearing in.
Yes, it's
in
more
vertebrate and
related
these
to
bones.
paleontology. So you're asking if it's better one or the other?
Yeah, so what I guess
Which
is
are the
that,
pro and the cons of the two?
yeah, so here you have one tool, and when if you have a hammer, everything looks like a nail, right? So
See,
that's sort of what's happening here, and I agree that it is better in some ways, but maybe there are other ways or other cases in which it's better to use a classic way.
well, in my context, I will not define it
I
classic,
agree that for species.
but I think they are really well interconnected, because if I don't have fossil, if I don't have the classic phylogeny, which is, relationship and dates, I cannot build mine, for example. So it's really important for the calibration. Maybe mine has a more resolution power. I can go deeper in the details and really see species, no species, most of the time. But if I don't have the context, the herd context, if I don't have the framework of the calibration, I cannot do it. So they are different. I think the paleontology framework, paleontology phylogenetics, it's course in history, and it was really important to set. But now for the details, for the little leaves in the tree, it's more important the genetics one. So I will not say that one is most important than the other, but it's like a progress story. They both contribute to the phylogeny.
You have to read a lot about how to establish homology. And because in a sense, when you build a data set, and you position in your data set, in a sense, you have a primary homology. You assume that position A1 of species 1 and species 2 is the same, is homologous. And you have to establish that. If that is wrong, that everything behind that is wrong. And for genes, very conserved genes, it's very easy to find that position 1 of species 1 is homologous of position 1 of species 2, 3, 4, 5, 6. In a morphological character, the species are very close related. Maybe, I don't know, you compare primates. Maybe it's very easy to establish homology of this character or whatever. But when you compare a bacteria
Yes, also.
with a human, then establish the homology of that characters is no.
Yeah, I'm guessing if it's at the species level, then the small differences will be easier to cut with DNA. But if you're trying to classify classes or orders, and
Well, I mean, classically orders is artificial. Doesn't
it
exist.
is artificial, but at the
No,
same
it's
time,
artificial. It's artificial.
Yeah, but if you
It's artificial.
classify
Because, I
them,
mean, you go to taxonomies, you expect one group and the other one to use that as homologous. I mean, have changed a lot. I mean, that's true. But
species is also.
But systematics is done, and we use these characters. Systematics is to test if taxonomy is correct or not. I'm not saying that is correct or not, but systematics is a continuous. Taxonomy is not a continuous.
And you think DNA is also continuous.
Well, we assume that it is true because we assume that gene is homologous to that gene. The problem is when you assume that gene is homologous to the other gene and is not. And you have a problem because you use the species 1 and gene A, you compare it with the species 2 and gene B. Have to be gene A and gene A for the same. You compare gene A with gene B, you are going to have comparison with peras con manzana or peras con garbanzos, no sé. It's different. When you have to compare, in a sense, you have to assume that are homologous. The characters are not homologous, not make sense to do anything. And that is the most important part. Doesn't matter you use morphological characters, any character, you can build a phylogeny with any character. That's no problem at all. But you have to assume that they are homologous. And at this time of the species, it's more difficult to find homologous characters. That is the main problem of of phylogenetic. And in DNA is a little bit more easier to establish homologies.
If I can add a last thing, I just want to say, because you told us that, for example, at a higher taxonomic level, it's difficult with genetics. But I want to show you the mitochondrial DNA that I'm using, and it has different genes, and some of them are really well conserved. So, for example, the COTS one, we use it exactly for this reason. But since it's really well conserved, it's difficult to go into the details, for example, species level, in which case we use for example, with the ATP8, which is really variable inside it. So, well, even if you have DNA, you have different region inside it, a different level of, diversity. And this helps you also to set the level at which you want to go deeper into the phylogeny. Just for closing. Sorry.
Yeah, this is the best example of homologous gene, because every species has only one mitochondria and one mitochondrial genome. When you have mitochondrial genome, you are sure that they're homologous. In genome, it's very difficult because you have many duplications during evolution. You have the genomes evolve by duplication. And duplication, and this gene works for one function, and the other one works for another function of duplicates. And then it's sometimes very difficult to find which one homologous A and B correspond to other species A and B, because sometimes you may compare the A from one to the B from the other species. And for that, it's so difficult to find homology. And this one is the best character you can use to reconstruct the phylogeny. The only problem that is that you have a lot of saturation. I mean, saturation means when you have many mutations changing the same position, you have saturation, because after a lot of time in evolution, you have in that position 20 changes, 30 changes, 40 changes, 50 changes. How you were able to reconstruct with the model 50 changes during evolution? In nuclei, you have very few changes in one position. I mean, you have very few changes in one position. It's very easy to reconstruct the changes having done here for the model. And that is the problem of this. That is very easy to find homologous characters in between species or among the species, but you have many changes on the position. Then it's very difficult to reconstruct the many changes. And you have many changes, it's very difficult to set the clock and time the clock. That is very difficult. And that is the main problem of this gene.
It's super interesting discussion, but I think that we have to stop it here. It's almost lunchtime. So thank you very much for all the speakers from today.